Actually, the next speaker is a good friend, a very, very bright guy, and actually what I would consider a real success story for otolaryngology, because otolaryngologists are an interesting breed because we interact with pediatricians, neonatologists, geriatricians, and everybody in between. And a lot of otolaryngologists like Mike Johns have ended up CEO of places like Emory. And Cliff is a great clinician, great researcher, became chairman at Case Western and is now essentially in charge of all the physicians involved with the Case Western Reserve University. We've kind of deemed that the vice president of severe combined pubonus, but it's actually a vice president role at Case Western, which is just, I'm saying it right, aren't I, Cliff? What is the actual title?
President, Chief Medical Officer.
Yeah, President, Chief Medical Officer. So quite a success story, but still, at the core, he's a neurotologist who's really good at what he does. So Cliff Megerian will come up and talk to us about Meniere's.
I think it was 1995 or 1996, so 20 years ago, Med-El was coming into the American market and invited a group to go to Innsbruck. I don't know if Doug remembers that. And I met Doug for the first time, and we had a great time there. It was a good group of people. And through the years, there's kind of a small group of us who have become, I'd call it our travel group, who are friends, like Doug and David Haynes from Vanderbilt, and Craig Buckman from North Carolina, and Alan Miko, and a whole group of us have, in our era, have stayed in very close touch. And I'm really pleased that each of us have kind of found a niche, and Doug has just done great out here. The other thing that's really fun is to kind of watch the circle of life. I remember in 2003 or 4, when I was new, I had come from Boston to Case Western. I was interviewing residents. There was this bright-eyed kid from Columbus, Ohio State, and God, we liked him. We wanted him to come to our program, but sure enough, he chose the University of Michigan because we would have ranked. And here I am now, these years later, and see what an amazing doctor Dr. Lorenz has become, and I get to hear him lecture and learn from him. It's really, I think, wonderful, and that kind of teaches a lot about how the most exciting thing to do is kind of be in the game long enough that you can be taught by people that one day previously were looking to you for education. What I want to talk about today is something that's been a passion of mine since I was a fellow. I did my fellowship at Massachusetts Meniere Infirmary from 93 to 95 after doing my residency actually at Case Western. And I was fortunate enough to be on the tail end of Dr. Hal Shuknick's career. He had stepped down as chair. Joe Nadal became chair, but he was active and one of my preceptors in my research. And he was always fascinated. And of course, I became fascinated. So why, when you have Meniere's disease, why do you lose hearing? If you go around the room, all of you think about it, we still don't really know why, right? People thought, well, the hair cells die, or gosh, maybe because of the hydrops, the tectoral membrane gets pulled off in a direction off the hair cells, and none of that really is figured out. We don't really know why hearing goes down in some people to be severe enough to require a cochlear implant, why in other people the hearing stabilizes, and other people, despite having a dead ear, they still have intractable vertigo. So I became interested in Meniere's disease, and I began working with a fellow named Bob Kimura, who in 1965, he and Hal Shuknick created the first animal model of hydrops where they plugged the endolymphatic duct in guinea pigs, and it essentially has been the mainstay. So I used that model in my research, I think I messed this up. Okay, so there you go. All right, so maybe someone could help me know which is the one to push. Maybe this one? There we go. Okay. So if you think about Meniere's disease, the fact of the matter is it's been a long time. Prosper Meniere in 1861 really proposed this notion of a triad where you have hearing, episodic vertigo, fluctuating hearing loss, and tinnitus. Prior to that, it was really thought to be some sort of epilepsy or brain problem. And he proposed really that the underlying pathophysiology was due to endolymphatic hydrops. He actually proposed that there would be distension of the Reisner's membrane and too much fluid within the scala media compared to the scala media, the scala tympani, the scala vestibuli. But it really wasn't until 1938 that Hallpike and Carnes, interesting, the same year, Hallpike and Carnes in England and Yamakawa in Japan got their hands on a cadaver, temporal bone of someone who had Meniere's disease. It's a really long, fascinating story, and those which I won't go into. But the bottom line is they're able to decalcify, section, and they found hydrops in both of them. So the notion of hydrops being actually part of Meniere's was really first in 1938. And it was 1965 that Kimura and Shuknik first did the guinea pig model. The problem with that, and why do we even need a model? We need a model because no one in this room can adequately tell for sure a patient why they lose their hearing with Meniere's disease. We know why you lose your hearing with chronic otitis media, because your eardrum's got a hole in it, your ossicles get broken down. We know why you lose your hearing with acoustic neuromas, because your cochlear nerve is getting crushed and losing its vascular supply. But we really don't know exactly the mechanism of losing your hearing in Meniere's disease or hydrops. And why is it important? Because if we knew, we'd figure out what we can do to stop the hearing loss. We kind of have an idea about the vertigo, right? We use diuretics, low salt diet. Some people use shunt. Worst case scenario, we can cut the nerve, but we don't have really any way of intervening on hearing loss. So the model is very important. You have a model that works. The problem with the guinea pig model, and I've spent years with it, it's very, very difficult to produce. I used to do surgeries every Wednesday morning for about 15 years producing guinea pig models. They are more difficult. They spend more time in the post-op unit than an ears patient in Germany. They literally have to have nursing for 3 days, blood pressure. It's unbelievable. And I became very discouraged, and I wanted to give up research. And I said, gosh, I got to find a mouse model. So what happened was, this shows you the power of the internet. It was 2005. I had just received my second sanction letter from the Animal Control Committee at Case Western that I wasn't properly providing, I think, entertainment for the guinea pigs in their post-operative period. I'm serious. That's how it was. And I said, I can't go on this way. I really can't. And so I said, gosh, there's got to be a mouse. I'm not smart enough to produce a mutation. So let's look through the Jackson labs and find out all the mutations they have and see if there's any statement that says hydrops or hearing loss. And I came upon this paper by Lorenz Depreux and Ken Johnson, who I now know, in 2004, where they're studying rickets. And they had a mutation it was a spontaneous mutation called the HepDuc mutation that has mutation in a phosphate regulating gene. And they used that to study rickets, but there was a small statement deep in one of their papers that was in mammalian genome that said vestibular dysfunction, hearing loss, and hydrops of the ear. So I said, let me get a hold of these. That sounds kind of interesting. And so my first job in 2007 was to start characterizing, seeing if these mice could be used. And here's the gene. It's a spontaneous mutation. It's X-linked, so it occurs only in boys. So what you have to do is you have to take a tail snip and do a PCR for the mutation. And let me have a pointer. Just have a... So if this is going to be a useful model, the animal can't be born with hydrops, right? It has to, and we didn't even know these are not otologists who wrote this paper from Jackson Lab. They're basically musculoskeletal specialists. So we have to get a hold. So this is a day 0. This is a temporal bone. This is a wild type mouse. You can see at day 0, the day they're born, they have a nice, proper Reisner's membrane with no dilatation. And sure enough, thank goodness, at day 0, the mouse that has the mutation, in other words, the X-linked mutation on the X chromosome with the HepDuc mutation and normal Y, they also have a normal Reisner's membrane. So here's a P10. P10 means 10 days after birth. At 10 days after birth, obviously the normal mouse has a normal, now you can start seeing beautiful architecture. Here's the Reisner's membrane. Here's the stria vascularis, the lateral wall. Here's the organ of Corti, tectorial membrane. Here you can see the spiral ganglion neurons. And sure enough, at 10 days, these mice all have fairly well positioned, no evidence of hydrops in the Reisner's membrane. And I was kind of stupid at the time. I thought that the only way really these mice, maybe if they were going to get hydrops, they were going to have obstruction of the endolymphatic duct. Thinking, it was kind of naive. So I looked at endolymphatic ducts. It was obviously open in the wild type, and it's open in P10. And finally, then we start seeing something. At P21, so obviously you let these mice live, you sacrifice them. P21 wild type normal, rhizos membrane, but look what you're starting to see in the rhizos membrane, the mutation. You're starting to see hydrops. But still, the endolymphatic duct is open. So this is happening in de novo, independent of endolymphatic duct obstruction. Look at P40, what starts to happen. You have such severe dilatation of the rhizos membrane that it's starting to become juxtaposed to the osseous spiral lamina here. And now as we get to P90, you can see again the wild type, which is here XY, normal, rhizos membrane, full complement of spiral ganglion neurons. And here's what's a very important thing for me to see. If you look very closely at the organ of Corti, obviously in the normal mouse, you have 3 beautiful inner hair cells, inner hair cell, 3 outer hair cells, rhizos membrane, really nice spiral ganglion. Here's our hydropic mouse at P90. Whoa, basically normal organ of Corti. Why are they losing their hearing? Look here, what are we seeing? Any medical students or residents here? What is this? What's the difference between this spiral ganglion? So we have a normal end organ. It's neural degeneration. So at least in this model, it started to point out some of the things that we see in our Meniere's patients, that the mechanism of hearing loss appears to be a neural problem, as opposed to other forms of deafness, which are a hair cell problem. So that was very interesting. And it's topographically localized. In other words, if you look at the apex, you're starting to see a clearing. Where do we start seeing our hearing loss first in Meniere's disease? Low frequency. Where's low frequency? The apex. And then if you see the mid portion and the basilar portion, they still have relatively preserved spiral ganglions for a period of time with the hydrops. So this is recapitulating perfectly the human condition. So wow, our conclusion was this is a pretty good model. You see basically spontaneous development of vestibular dysfunction because these mice start twirling at about 25 days. Cochlear dysfunction starts right around the same time. Endolymphatic hydrops starts actually before. So clearly there's something biochemical going on. It's not the act of the hydrops itself. It maybe the hydrops is an epiphenomenon to some biochemical issue going on. And then there's neural degeneration that sets up. So conclusion of this part is that this mouse, and this was published in Hearing Research in 2008, and this is important because, we as clinicians, we publish in laryngoscope, otology, neurotology, but if you really want to hit as a clinician your research colleagues, it has to be in JARO or Hearing Research. So this obviously took like 70 revisions. We finally got it in. What the beauty of this mouse is you can voluminously reproduce a platform now to study hydrops and how to truncate hearing loss. And obviously, you've got to have the capacity to be able to know when you have 2 little babies, to be able to do a PCR from their tail vein, from their tails, and figure out with PCR which is the mutated one, because you can't tell when you look at them when they're born. So my hypothesis then is if you have hearing loss and it's neural-based, there's really only 2 things that can be happening, right? Necrosis or apoptosis. Apoptosis is programmed cell death. So I thought, why don't we look into the possibility of the mechanism is apoptosis? Because if it's apoptosis, then we potentially could come up with strategies of stopping the hearing loss, right? Why is that important? It's important because we're way behind. Right there, we know in Meniere's disease, we're basically dealing with a neural phenomenon and a spiral ganglion degeneration process that appears to be the principal area. What is one of the biggest areas of drug development in ophthalmology? Glaucoma. What is glaucoma? Glaucoma is essentially increased pressure in the anterior chamber. And what happens? Retinal ganglion degeneration. Essentially an analogous disease process. And there's companies that have been built around this, like Alcon, where you have tons of drops and drugs that are used to stop the apoptotic process or stop the inflammatory process that develops. We don't do that with our patients, obviously. We're far behind. Our closest thing we do is starting to squirt steroids in the middle ear. But imagine if we knew what were the time points along with neural degeneration happens, and we could start, put a tube in someone's ear, give ear drops that have, let's say, an agent that stops one aspect of apoptosis, if apoptosis is correct, and we could preserve hearing. And that's, I think, the future, really, of managing this disease. So I think the first step was for us to evaluate the hypothesis that apoptosis is really what's going on. Here's the hypothesis that spiral ganglion degeneration affects mouse, at least in part, undergo apoptosis. So we take mice, P60, P90, P120, and we decide to take downstream regulators of apoptotic cell death, caspase 3, 8, and 9. So we do basically analysis of activated caspase using PCR techniques, quantitative PCR techniques, and then finally do a tunnel assay, which is a diagnostic of apoptosis. So if you look at P90, here's a normal mouse, for example, and here's a mutated mouse. You could see the density of the neuronal population has significantly declined, and you can see in this group, you're starting to see significant, what you see in apoptosis, you start seeing granulation within the nucleus, and these granular spots start developing. Now, this is a histological, and it's not really accurate, but at least tells us we're on the right track of thinking apoptosis is the pathway of neural degeneration. And then we do corrected spiral ganglion cell counts. And interestingly, just like we saw in the previous slide, that if you take P90 and P120, here's the normal mouse, here's the normal density of spiral ganglions in the apex, and you can see that the normal mouse has about 1,200 cells per square micrometer, whereas the apex in the evolved mouse has gone down significantly, less significantly in the middle, and it's almost normal at the base. But then if you go to 120, you're seeing further deterioration in the apex, further deterioration in the middle. You're starting to see deterioration in the base. And this was published also as well. So using quantitative PCR analysis, then you take out that tissue, and we're going to measure using basically a platform of quantitative PCR analysis. And this is really cool. You're starting to see that at P21, you have caspase. This is elevated compared to control. Okay, so this one is already almost 2 times the normal at P21. You see at P40 how much increase in caspase 8, which is a principal mediator of the pathway of apoptotic cell death. So this is starting to line up pretty nicely. Now, this is really cool. We're looking at immunohistochemistry, and we have a wild type, and you can see that in the spiral ganglion, we're getting really no enhancement of caspase 8, but if you see in P90, look how you're starting to see the neurons specifically are starting to light up here. Okay. Similarly, caspase 8, much more. Here's the control mouse at P60. You see here's the spiral ganglion. You can see these neurons are really starting to express a caspase 8. Similarly, caspase 9. And this is really cool because it shows that if you look at the tonotopic enhancement, here's P40. You can see the enhancement is first in the apex. Looks like the base is pretty clear. Then I'm sorry. This is the wild type. Here's the mutate, and you can see that there's significant enhancement predominantly in the apex, not at the base. But if you go from P40 to P90, which is, if you will, when a person goes from maybe having Meniere disease year 1 to Meniere disease year 7, look what's happened to the base. It shows the progression of the disease involving the whole. And here's the tunnel staining, which is it's a process by which you can look at cells specifically, and it's really pathognomonic of apoptosis. So, and here's the path. Here's our theory, really, and it's not really theory. I mean, apoptosis and neuronal cell death is not ours. That's been around for a while, but we're just suggesting that's what's going on in Meniere's. is that here's the hair cell. We know the primary neurotransmitter in the hair cell for type 1 neurons is glutamate. Glutamate is released. Our theory is with the hydropic phenomenon that somehow the metabolism of glutamate is impaired, and it sets into motion through the NMDA receptor a series of events which start activating caspase 8 through caspase 3, which leads to DNA degradation within the neuron and cell death. So that's kind of our story. That's what we think. So next step, looks like I have 10 minutes left. The next step is, wow, this is great. So we think we have a pathway. We think that we have a mechanism of potentially intervening in neuronal preservation. So let's look at some neuroprotection. Let's kind of talk about that. But before that, how many of you in the room order Ecogs on your patients? So why I do it, and I did it for years even before I even got interested in this. We do that because there was a suggestion that electrocochleography is indicative of endolymphatic hydrops. But has there ever been a real study? We can't sacrifice our patients to check if they have hydrops. It was a mathematical assumption. There's never really been a study that really showed is an elevated SP-AP ratio truly consistent with endolymphatic hydrops. So I thought, it was a great opportunity to do that. They tried to with guinea pigs, never really worked. There's really, if you look to the literature, there really is no publication. So let's do that. So we took 42 normal mice, 26 mice with hydrops, ABR and Ecog recordings at P21, histological analysis, measured the degree of hydrops and the degree of hearing loss. Here's the results, very quick. So here's the hearing thresholds, the normal mice, and you can see, and this is about what you're going to get in ABR testing, normal mice. Here's the degree of range, but you're going to get around 40 to 50 in terms of a click. And here is the range at 21 days. Remember, you can't test hearing in 19 days in these mice. They have to be at least 21 days because they die when you give them the anesthesia. You can start already seeing elevation of the thresholds. And this is really cool. Here's the bundling of the SP-AP ratio distribution. In the normal mice, it comes down around 0.3, and in the mice who have, at 21 days, the Ecog shows around 0.5. That's kind of consistent. For those of you who order Ecogs, what do you use as your 0.4, 0.5? So this is really lining up. But here's the thing that was really cool. Unfortunately, there was absolutely no relationship as we started measuring over time to the relationship of the degree of hydrops and the SP-AP ratio. And also, it did not really have any relationship to the degree of hearing loss. It's flat, as opposed to going like this if there was a relationship. So our conclusion is elevated SP-AP ratio associated with the presence but not the severity of endolymphatic hydrops. So that was published, just came out in also in hearing research, actually 2014. And it has much more background. And that's the first author of that is Melke. So the next question, which is I think where the money is, can inhibition of apoptosis somehow protect spiral ganglion degeneration? So we got a great idea using silencing RNA, which yet hasn't been approved for human use, but the concept of silencing mRNA, siRNA, blocking the mRNA of deleterious proteins. In this case, we chose, what do we choose? We choose caspase. It's a 20 to 25 nucleotide long double-stranded RNA and specific gene expression silencing. So the idea is something like this. It goes in, it essentially blocks the mRNA that's coming out that is coding for that deleterious enzyme, which in this case is caspase, and hopefully we could stop the end product. So we took 3 different groups. We had a vehicle, which is really not just giving saline. We had an siRNA against gene X. I can tell you it was a caspase-related gene. The reason we don't write this here is we had a partnership with a drug company called Quark, who was making the drug for us. And then here's the active agent. And here's the times that we would topically administer. Interestingly, we didn't inject it. We just put it in the ear canal. And the idea is it's so small, it just fused into the middle ear. So here is the outcomes. P90, You can see with vehicle, you see everything that we want, what we expect to see. Horrible hydrops, complete degeneration of the spiral ganglions, but with the agent, we're able to get neural preservation. Similarly, when you take a close-up of the basal the basal part of the spiral ganglion, you're having in the vehicle, that means the control, significant neural degeneration. Here we're getting some preservation. Here is the spiral ganglion cell counts. Here is here's the vehicle. With the vehicle, gene, and then gene that's inactive, gene that's active. You could see in the apex where we should see significant deterioration, right? Compared at P90 that I told you before, we're seeing some relative preservation, not complete preservation. Here's complete preservation will be up here, but relative preservation using the gene. It's pretty exciting stuff, right? I mean, theoretically, we would be able to have a product like this for people with neural degeneration or Meniere's related hearing loss. Similarly, here is the hearing preservation. Here is the control. You can see over time, postnatal day 20 all the way up to postnatal day 90, you see a significant deterioration hearing, and here is the preservation with the active agent. Ecog results show no real difference. So what is that? Why is that important? That's actually what I was hoping for. Basically, what it's saying is that the substrate, which is the dilatation of the rhizom's membrane and the hydrops, is still there. But somehow we're protecting against the downstream effects of the hydrops, which is the neuropathology. So this is preliminary stuff. So apoptosis gene silencing appears to confer otoprotection in the FEX mouse. These are the kind of things that will hopefully lead to potential avenues of pharmacological treatment to prevent hearing loss in our Meniere's patients. So this was just recently presented at a Meniere's conference. This is the team. It's a big team. Yin Zhang, who actually we recruited from Jackson Lab, who worked with Ken Johnson originally on that mutant mouse, came over to Case. Here's Cameron Wick, who's his chief resident. He's going down to his neurotology fellowship at UT Southwestern, and I just signed him to come back. Maroon Saman, who knows Mark Lorenz, trained at Hauser Institute. He's joined our faculty and other members of the lab. Here's a bigger group of the researchers. We have 5 funded NIH researchers, PhD researchers in the ear section of our department, and led by Kumar Alagramam. Here's Brian McDermott, who's a zebrafish guy, and we've just added 2 more. But these are the members of the team that were involved in this research here. So they're obviously very, very involved. Why is this stuff important? It's kind of boring for a group who's here to study surgery. But in reality, I really believe there's a significant homology between glaucoma and Meniere's. I think if you read the original work done by Prosper Meniere, he even suggested that it is consistent with or an analogous disease process to glaucoma. And now, as we know, certainly glaucoma, there's surgeries and there's shunts, but ultimately the mainstay is drug therapy to preserve vision. And these are the kind of things I think that may happen in the future. So thank you for listening. Hope I didn't put you to sleep. Happy to answer any questions. Yes, sir. Well, so I firmly believe diuretics are very useful. I use it in my practice all the time. I believe that what you're doing is you're somehow minimizing the hydropic load, obviously. But how do I explain the fluctuations? That's a tough one. I think that I think what's going on with the fluctuations is during a severe fluctuation, which is accompanied by a vertigo spell, you're actually having a membrane break. You're having a membrane break, and you're having admixture of endolymphatic and paralymphatic fluid, and it's really poisoning, if you will, and temporarily shutting off the transmission of the hair cell. And then when it finally seals and heals, you oftentimes restore hearing, but to a lower extent. I think that's the fluctuation. I think the progressive slow decline is the apoptosis. Yes?
Are your mice vitamin D deficient, or they have a deficiency in their vitamin D metabolism? Do you think there's a role for that?
Yeah.
In the model?
Yeah, so we just presented actually one, the American Otology, Cameron won it, the American Otology Society Research Award at the last COSM was doing just that, supplementing these mice with vitamin D. I think you're asking a very fundamental question. Are these mice vitamin D? And the answer is yes. And so could that have something to do with it? And the answer is probably yes in these mice. That if you look very closely at the bone of the OTA capsule, there is some deterioration of the bone, which begs the question, and people have really raised this in the past. In other words, some people believe Meniere's disease is an OTA capsule issue, which then leads to a problem with maintaining homeostasis within the fluid spaces. So we did a study where we supplemented, it's coming out in Otology and Neurotology. We didn't have a big impact. We saw some improvement in the histology, but not a dramatic. We basically used vitamin D supplementation, calcium, really those T, vitamin D3 and calcium. That was at last year's COSM. But I think that's potentially the thing. In other words, could Meniere's disease really be an OTA capsule issue? And could it be, in other words, the treatment could be involving things like that? And what we're seeing is we're paying attention to the membranous labyrinth, but in reality, it's an OTA capsule issue and potentially related to a bone metabolism issue. There's a whole bunch of people who believe that's going on that in our field through the years, even way back to Shambaugh, believe that. Yes.
Do you have any comments on what forms what a lymphatic shunt actually does, and tie that in with this article of cutting
Yeah, I mean, so endolymph is produced with the stria vascularis, the dark cells of the stria vascularis, and some of the dark
Okay,
cells.
Cliff,
Yeah.
I'm going to call it timeout here because that's case number 1. We're going to talk about that. And so we're going to pause you.
Okay.
We're going to let you restart.
All right.
Because I got a feeling that people probably want to make sure that